Okay. Father, Son, and Holy Spirit. God, grant me the serenity to accept the things I cannot change, courage to change the things I can, and the wisdom to know the difference. The Father, the Son, the Holy Spirit. Amen. Well, you guys are too young to remember any of this. You weren't even around. Most of you weren't even around, right? Anyone alive during 9/11? Okay, Kate, a couple of you. Okay. So I was up on Chestnut Ridge, just about 8 miles that way, when the Shanksville plane flew right over the property where we were actually staying on that day. So some iconic images. And heard a couple of nice quotes over the last couple of days. One was from Abraham Lincoln. When the people from the up north were upset that it became obvious near the end of the Civil War that he wasn't going to punish the South more. And he quoted the gospel where he said, Jesus tells us we are to pray for our enemies. And he said, no, I believe in destroying my enemies. And Abraham Lincoln responded, he said, well, if I make my enemy my friend, haven't I destroyed my enemy? If my enemy becomes my friend, haven't I destroyed my enemy in a certain sense? And there was another good quote from Thurgood Marshall, who was a justice on the Supreme Court. I do believe he was involved with the civil rights movement back in the 60s. When he was named to the Supreme Court, he said something along the lines of that laws are good at certain things. They can break down walls, and they can make things better. But in the end, in the final analysis, it takes people willing to reach out to one another that actually builds bridges and connections. So, 9/11. Anyways, that's part of my history. Oh, speaking of history, here's the history of cells. Lysosomes. Hey, we talked a little bit about these on Wednesday. They're membrane-bound vesicles, structures, that contain digestive enzymes. These arise from the Golgi apparatus. So enzymes are proteins. They are oftentimes proteins that break down molecules or make reactions happen. Because they're digestive enzymes, they keep them hidden within these little membrane-bound vesicles so that way the enzymes don't digest you. So anyone ever seen the movie Spaceballs? Spaceballs, anyone? Okay, Pizza the Hut. How did Pizza the Hut die? He got locked in his car and he ate himself to death. That's right. So we don't want to be like Pizza the Hut. So these digestive enzymes are locked away inside vesicles. What are their jobs? Number one, they can break down old organelles to repair their component parts. Like this old mitochondria. I just can't do it anymore. Well, it's time to break you down and recycle you. So they'll break these old organelles down, digest it, use the parts, recycle, make something new. That's what they do. Our bodies are much better at recycling than we are. Much better. They break down and digest other cells ingested by the cell. So some cells we know, some organisms, actually, they engulf their food, they surround it, pull it into the cell, and then that food is then digested inside the cell. It's a good example of what's called endocytosis, which we'll talk about. Endo, bringing into the cell. All right, lysosomes. We're just introducing that. So, if a protein is a digestive enzyme that ends up for the lysosome, where is synthesis for that protein started? Always in the run. By the way, remember, zombie is a second language. They're having courses, or masked zombie is second language. I'm taking that class, I'm just kidding. Cytosol, right? Cytoplasm, okay. Where does it go next? If it's bound for a lysosome, it's going to then be, the synthesis is going to be finished in the rough ER. Thank you, good, all right. Hey, we don't have a quiz on Monday. Okay, so, components of all cells. Plasma membranes. Their main job is to create a boundary that separates the inside stuff from the external environment. They're selectively permeable. Right? That means some things are allowed in, some things aren't. Some things are allowed in, some things aren't. It's called having a boundary. Okay. This allows necessary molecules that keep the molecule alive. There's a Queen song, keep yourself alive, keep yourself alive. Let in what you need to let in to keep yourself alive, right? So they allow necessary molecules to come into the cell. They keep the harmful ones out. So that's the entrance. But it works both ways. They also allow waste products to get out of the cell. If it's toxic, take out the trash, right? Take out the trash. Take it to the dumpster. Get it out of here. Send it to China. They'll deal with it. That's where most of our garbage goes. Other countries. We ship our garbage to other countries. Why? They have more room than we do, I guess. I don't know. But anyways, that's a topic for an ecology class. So, waste products leave the cell and keep what you need inside the cell. So it works both ways. Now, oftentimes the plasma membrane is described as what's called a fluid mosaic. Fluid mosaic. So fluid. First of all, it's not static. It's not rigid. It's mobile. It's flexible. It moves. And it's not uniform like a plastic bag. It's a site of constant exchange of molecules, and there is a wide variety of different proteins that are present in the plasma membrane. That's why it's a mosaic. It's not the same throughout. And in a biological system in general, we have to remember there's like a principle of biological systems, that biological systems that are homogeneous, that are all the same, are intrinsically unhealthy. Biological systems that are comprised of all the same thing, homogeneous, they're unhealthy. Healthy biological systems have a wide variety of life forms, structures, etc. So the same with our plasma membrane. Now, some of the things that are found in this plasma membrane are these phospholipids. How many fatty acids in a phospholipid? 2. Good. Saturated fatty acids. What's that mean? What? So every carbon is saturated with it's saturated with hydrogens, right? There are no what? Tristan, no double bonds between carbons here. Beautiful, good. So if the plasma membrane is made of phospholipids, predominantly phospholipids that are saturated, this makes it less fluid. Because those fatty acids, what's the interaction that happens between non-polar molecules? We call it a blank interaction. Hydrophobic interaction, right? So we got a lot of we have lots of hydrophobic interactions between these saturated fatty acids, push the water out, less fluid. If we've got more unsaturated fatty acids, then that makes the plasma membrane more fluid. More fluid. So quick question. You're a polar bear. Who wants to be a polar bear? Okay, Deshaun, you're a polar bear. You want to hunt him? He's like, I'm hungry. There's a whole room full, and he's kidding. Anyways, okay, so you're a polar bear. It's cold. Are your plasma, and then who wants to be a, I don't know, parrot? Parrot. Quattro's a parrot, okay. All right. Parrots are cool. All right, so living at the equator. All right. Whose cells are going to have more unsaturated fatty acids? Deshaun's or Quattro's, and why? Think about what happens. We talked about some specific properties of substances that either have a lot of saturated fatty acids or those that have a lot of unsaturated fatty acids. So think about that. Polar bear. Or parrot. Who's going to have more, whose plasma membranes will contain more unsaturated fatty acids? And why? Why is it important? Okay, Aiden? Okay, polar bears because of? Okay, so the plasma membranes of the cells of the polar bear are likely to have more unsaturated fatty acids because at cooler temperatures, they're going to remain what? What's that? Warmer, or they're not going to become this rigid structure. That's right. Everybody get that? So remember, a substance that's comprised of a lot of saturated fatty acids, what happens to it at room temperature? It is, in what form is it? Solid or liquid? Solid. We call it a fat, right? So something that is solid at room temperature, we call it fat, and that's because it has a lot of saturated fatty acids. Well, when you're swimming around, you're walking around the Arctic Circle, it's cold. Higher percentage of unsaturated fatty acids will allow that plasma membrane to remain more fluid. Okay, so Jashawn, if you really want to be a polar bear, hit that olive oil. Start hitting that olive oil today. Get some of those unsaturated fatty acids in you, all right? Okay, cool. So, phospholipids. 2 fatty acids bound to a glycerol with a phosphate and a choline part. Their function is to form plasma membranes. Now we've got all kinds of eukaryotic cells, has lots of membranes. And they're not identical, okay, but the membranes share fundamental structural features. They all have a phospholipid bilayer. There are transmembrane proteins. We're going to talk about those very shortly. There's an interior protein network. By the way, you'll be wise to know these cell components, right? Know the cell components for Monday. Just letting you know. And cell surface markers. That's these structures right here. Cell surface markers are part of transmembrane proteins. So we're going to see that 2 and 4 are kind of connected. So let's talk about the phospholipid bilayer first. There's our proteins, interior protein network, and then the markers. So let's talk about the phospholipids. We've talked about this before. The primary structural component of every membrane. So if you see a membrane, think phospholipid. Here's a membrane, phospholipids. There's a membrane, phospholipids. Old MacDonald had a membrane, E-I-E-I-O, and I know that it had phospholipids, E-I-E-I-O. Anyways, is this being recorded? Yes, it is. Okay. 3 cups of coffee, man. That's all you need, and a lot of Jesus. Okay. So it has 2 properties. Amphipathic means it has 2 properties. We got the polar region up here. Why is it polar? It's not just polar, it's charged, right? We got phosphate, choline. We got all kinds of polar-y things going on, which means this region can really interact with water, hydrophilic, nonpolar tails, hydrophobic, it's Golem. Oh, I didn't put him in there. Hates water. Okay. So we've said this before, you dump a bunch of phospholipids in a beaker, it's going to form a membrane. On its own, major advantage, once you make the phospholipid, ship it out to the membrane. Once it gets in place, boom, you got it. No more work. So here's a picture There's a cell up here. So this is the cytosol of cell 1 up above. Here's the cytosol of cell number 2 down below. And then here we see the plasma membrane. So the orange, if you can see the orange, it's kind of like a dreamsicle with dark dreamsicle-y stuff in between instead of vanilla ice cream. But anyways, so the orange, these are the polar heads. And then you see the dark region in between, that would be the tails. Here's plasma membrane of cell 1, plasma membrane of cell 2, and then here, this is that little sliver of extracellular fluid in between these two cells. This would be where your oxygen perhaps could come from, or your glucose could move from this little sliver of fluid here. This, my friend, contains the things that keep you alive. Stay thirsty, huh? All right. Let's do this. Transmembrane proteins. So that's the plasma membrane. Plasma membrane. Second, transmembrane proteins. 6 of them. Boom, 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 boom. Yep. 6 major classes. We'll talk about each of these in turn. Notice they're called transmembrane. So it's a protein fixed in the plasma membrane, and it stretches across the whole width of that membrane, right? So we've got, notice there's going to be a region that's exposed to the extracellular fluid outside the cell and the fluid that's inside the cell, the cytosol. Trans means across. Transatlantic flight. Trans-campus run from Benny to the refectory. I do it because I have to. Okay. So these membranes, they have 2 domains. So domain simply means a region of the protein. Okay, who can give me an update here? What, this is a protein. So this blue corkscrew looking thing, what do we call that? What level of structure is that? The blue corkscrew? Secondary structure. This is an example of a alpha helix. Comprised of what? What are the monomers? We've done this drill like 50 times. Amino acids held together by what? Peptide bond. By what process? I'm so happy. I'm gonna cry. Not really. Okay. So we're gonna have polar domains, which are gonna be in the red, and we're gonna have non-polar domains. Notice, now this is where it's really important to be able to identify what the R groups are and what kind of function, what kind of property they have. Polar domains, boom, boom. Interacting with the extracellular fluid. It's happy out there. Or the intracellular cytosol. It's interacting with these water-ish regions. It's also interacting with the polar heads on the phospholipids. Okay, whoops. So these are both aqueous environments. They're not watery, but they're watery. You know what I'm saying? It's not like there's an ocean flowing in between cells, but there is water there. Which means we're going to find amino acids, polar non-charged, and our charged amino acids. Those amino acids that have those R groups, they fall in those categories. You're going to find them in these regions of these proteins. That should make sense. If they can't interact, if those R groups can't interact with water, you're not going to find them there. I mean, there's exceptions, but in general, that's where you would find them, okay? The nonpolar domain is in here, where the amino acids are interacting with these very non-polar tails of the phospholipids. So remember, it's now at this point, it's the R groups of the amino acids that are interacting here. It's the R groups, okay? Not the carboxyl at the end, not the amino at the other end. They are busy forming peptide bonds. So they're kind of out of the picture. Or they're also forming the hydrogen bonds that help make the secondary structure happen. All right, anyways, I digress. So if amino acids in this blue region are interacting with the nonpolar tails of the phospholipids, that means that these amino acids are going to have R groups that fall into what category? Non-polar. Highly lipid, it's a high lipid environment, non-polar amino acids. All right, this is a big concept here. Circle, circle, star, star, square, square. All right. All right. Transporter. Let's start going through these one at a time. So they're all going to have these properties. Polar region, non-polar region. These allow molecules to enter or exit the cell. We're keeping this real simple. There could be water, no. Enzymes. They are oftentimes embedded in plasma membranes. There's enzymes and proteins inside of your cells as well, but we're talking about these ones here. And they carry out different reactions. An enzyme makes the rate of a reaction go faster. A lot faster. All right, so this is a membrane. We're talking about transmembrane proteins here. Just remember that. This is embedded in the membrane. Cell surface receptor. This allows for the interaction between the receptor in blue and the specific molecule in green. So what happens, this could be a hormone, for example, a hormone. There's a hormone we've talked about numerous times, insulin. So maybe this is an insulin receptor, this blue structure is an insulin receptor. Insulin comes along, there's insulin, binds to its receptor, and then this little signal here, this isn't Bluetooth. First of all, it's not blue, and it's not a tooth. But that's just showing that there's some sort of a signal that's being produced inside the cell. All right? Change inside the cell. This is one of the main ways, actually, these are the ways that the cell on the inside communicates with what's happening in the outside world through these proteins. All right? You guys ever, when you were kids, how many actually has ever done the thing where you take the shoestring between the cans, you oil the can down, you guys didn't ever do that. You did it? It actually sounds like you're talking in a mask. That was an old phone. He wanted to pretend. Anyways, okay, whatever. All right. Cell surface identity markers. It's a transmembrane protein that has a polysaccharide tag. Poly, many, saccharide. So what are these structures then? What are these? What are these? Sugars. Da 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 da. Oh, na 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 na. And this polysaccharide tag, its whole job is to make sure that when your immune system, the cells from your immune system go around, that they identify, up, this is Marie, up, this is Marie, up, this is Marie, not Marie, and then they unleash chemical whatever. All right? That means it's you. Polysaccharide tag. That's its job. Cell-to-cell adhesion allows specific interactions between extracellular regions of the cell, holds the cells together. There's a region in that holds your heart cells together called the intercalated disc. Holds those cells together so that way every time your heart contracts or expands, it doesn't blow apart. These are very important. Cell-to-cell adhesion. And finally, attachment to the cytoskeleton. So this would be one of those cytoskeletal proteins. Vast network going through the cell. They're going to be attached to these proteins. Helps the cell maintain its shape. All right, helps the cell maintain its shape. So 6 transmembrane proteins. They're all proteins. They all perform different functions in the cell. So recall that the primary function of the plasma membrane is to form a barrier, semi-permeable barrier that generates and maintains the intracellular environment, helps maintain those conditions inside the cell that keep your cells alive. Keep my cells alive. Keep my cells alive. I can't sing, but I don't care. Anyways. And keeps the foreign or unwanted molecules, keeps it from entering the cell or kicks them out. So it has to be semi-permeable. So we've said this. It allows nutrients and those that we need to come in, prevent harmful molecules from entering the cell, keep stuff in that needs to be in. So it's semi-permeable. Semi-permeable. Even necessary things are only allowed to come in at certain times. Like sodium. Concentration of sodium is much higher outside the cell than it is inside the cell. Much higher. That sets up a concentration gradient. So sometimes when the cell needs to do work, it allows sodium then to move in. And sometimes when it does that, it moves other things in as well. So, anyways. So what is it that determines if a molecule can enter or exit the cell? A couple properties. Size of the molecule, polarity of the molecule. Remember, I said way back when, being able to identify, hello, are you there? Yes. I'm sorry, what are you? You're polar. You can't come in, okay? So this helps determine whether or not something interacts well with water. And it also tells us how easily it can enter or exit cells. Very important. So, small, non-polar molecules. Let's take a poll. Poll. Who thinks that small nonpolar molecules can easily enter the cell? Sean, we got 1, 2, 3, 4. Who says it doesn't? Okay. Deshaun, why can't it? Why do you think it can? Okay, because it's nonpolar should be able to slide right through. Okay? How about, that sounds reasonable. How about a no? Where's a no? A reason for no? Okay, so maybe it's going to be repelled by the polar heads. Because polar and nonpolar, maybe they don't like each other. Get out, okay? So, that's something we need to think about. What region of this membrane might actually keep things out? Okay. Small nonpolar molecules easily pass through plasma membranes. It's like, this is going to be a bad analogy, but if you've ever read any of the gospel passages where Easter Sunday, they're all in there, the door's locked, and Jesus comes in. He's like, no stopping me now, man. I'm really nonpolar. He's resurrected. He goes where he wants to. So small nonpolar molecules go where they want to go. They just can pass through. So the polar heads don't repel nonpolar things. Okay? But that's an important concept to hang on to. We're gonna see that. Two really big examples that I want you guys to lock and load into your brain, be able to pull it out when I ask you. I'm gonna say, Antonia, what's a nonpolar molecule that can easily pass through a plasma membrane? You better be able to tell me oxygen and CO2. I want you to have it there. We need to know that. Okay. Large nonpolar molecules. Am I clear this morning? Am I clear? Are you catching me? Are we following? Okay, I just want to make sure. So I'm speaking fast. I apologize for that, but we gotta move a little quicker here. So large nonpolar molecules. Yes. Okay, Deshaun's shaky. No. I don't care. You don't care enough to put your hand. I'm just kidding. Okay. They freely move too for the most part as well. So, a good example, steroids. Steroid hormone, make you big and strong. I've been to weight room for last 24 days straight. My bicep is now 34 inches. Whatever, okay. Where does that come from? I'm like, I'm not all here, so large nonpolar molecules, they easily pass through, move in and out of cells. So steroid hormones, like cortisol, like testosterone, estrogen, in and out, easily. Progesterone, they move in and out of cells very easily. Small polar molecules. John. They go all the way through the plasma membrane. They're like, they slide by. They just go right through. Yep, they squeeze between. It's all concentration dependent. So if there's a higher concentration outside than inside, there's a diffusion gradient that would draw them through the cell, sort of push them through the membrane, and they sort of go through there. Okay, good question. Now, steroids aren't going to move through the membrane as fast as oxygen, that's for darn sure, because of the size. And there are some polar groups on some of those steroids, but it's still, it's going to go right through. All right, doesn't need any help is what I mean. Small polar molecules. Due to the small size, some water can pass through. Some water can pass through, but for the most part, small polar molecules do not pass through. Gandalf is standing at the bridge of Khazad-dûm and says, "You cannot pass," to the Balrog. Large polar molecules, no way, can't do it. Nope. Cannot pass through the plasma membrane. They hit the non-polar regions. That's what tells them they can't go through. So there is a region of the plasma membrane that serves as sort of the gatekeeper. It's not the polar heads, it's the non-polar tails. So, amino acids, glucose, cannot pass through the membrane. They can't do it. They hit it and they stop. They need help. I'm going to get through with a little help from my friends. Gonna move through the plasma membrane with a little help of my transmembrane protein friends. Okay, they can't do it. Amino acids, glucose, 2 examples. Small ions, forget about it. They can't do it. Can't do it. They can't pass through without a transport protein. And I said earlier that ion gradients are used as an energy source. We'll see how that works later. So, sodium, potassium, chloride, calcium, hydrogen ion, all of these are ions, cat or anions, and they can't pass through the membrane without having some sort of a transporter or a channel that allows them to pass. Okay. So what about transport? So need to know what those molecules that can pass, those that can't pass easily through. So now we're talking about how do we get things from one side of the membrane to another. So there's 2 primary ways. There's passive and there's active. There's passive and there's active. Passive transport. Simply stated, passive transport. No input of energy is required. And then here's the big one right here. Substances move what's called with or down a concentration gradient, which means it moves from high concentrations to low concentrations. Ready? Are you ready? Here we go. Passive transport. Right there. Just did it. What happened? It's moving from a high region in the room to a low region in the room. That's all it is. Let me do that again. You think it's going to go up this time, Abigail? Is it going to go to the ceiling? Let's try it. Not now. How can it get to the ceiling? If I give it some energy, right? That's called active transport. All right, but let's do this real quickly. So that means passive transport. So can oxygen simply move through the plasma membrane? Yes. Wait, Antonia? Yes. It can just move. Woo. So can it pass through the membrane? Yes, it can. If it can, then it's going to move from a region of high to low. Which means on the left here, it's going to move what direction? Right to left or left to right? Left to right. What about over here? Right to left. Follow the numbers, okay? That's it, passive transport. If it moves from a region of high concentration to low, that's it. That's the primary way you know if it's passive or active. Second, active requires the input of energy to move substances up or against a concentration gradient. Okay? It uses energy to move a substance up or against a concentration gradient, which means it's moving from low to high concentrations. Low to high. Because oxygen is nonpolar and easily passes through membranes, it's always going to move passively down. Always, always. Oh, are you serious? Oh, wow. Hey, how about that? Well, I guess, okay. I thought I had more slides on there. So, for the quiz. For the quiz. You will be wise to go back over amino acids, R groups. What are they, polar, nonpolar, or charged? Go back over those. You need that information. It's important. You got to have that. Quiz yourself, flashcard yourself, whatever you do. Know all the biomolecules, right? So everything starting from proteins, the biomolecule classes, name of polymer, name of bond, name of the monomer. Know it. Be able to identify that. Different levels of structure for proteins, right? 4 levels of structure. Be able to identify the biomolecule type if I put a picture up, like a picture of a disaccharide, or if I put a picture up of a fatty acid, you should be able to know that's a fatty acid. That's a lipid. If I put a picture up of phospholipid, triglyceride, polysaccharides, nucleic acid, whatever. Be able to, that's a nucleic acid, blah, blah, blah. And then pretty much all the questions, which I know you guys have been completing those as you go along, because we're now up to we didn't do fixed law, so question, well, up through the bottom of page 6. The question's at the bottom of page 6 in the study guide. So basically, all of page 3, 4, 5, and 6, those questions. All right, but you guys have been doing this studying. This should be a little bit of review, because I know that we didn't wait until the weekend before the quiz to start going over these. So, awesome. We're going to pick up here on Monday. Remember, the quiz goes live Sunday at noon. It's live until Monday night at 10. Don't forget, we're going to keep hitting these. Yo. right, I'll get the rest of these. These are mine. You don't think I can do it, that's the problem. You don't think I can do that. Don't know what that is? Well, that's it starts at 7. Yep, Wimmer Lawn. Default microphone. Yeah. Dee-dee-dee.